Can I get a whoop up? Can I get a whoop up? Set weakness. Set minus a minus. Can I get a whoop up? Cuenca. Cuenca. Hey, Cuenca. You hear me, sweetheart? You gotta put your mic on. Hey, are you in my classroom? Cuenca, you in my class? Hello? Maybe my volume's screwed up. I can't hear you. Oh, your mic doesn't work. Oh, that sucks. Sucks. What's up, Manus?
Good morning, Mr. Pope.
Morning. Oh, Amarai's got the soft copies. I mean, the hard copies. She's got the hard copies, so she's in a better spot than everybody else. Were you able to print these documents off yourself, Manus?
No, I wasn't able to. So I just did them on paper. Like I did them on paper.
Fair enough. Fair enough. There should have been several you couldn't figure out. Like the first one on the Heredity page, probably something you're just like, what is that crack pipe? I don't know what that is. Did you either one of you guys figure out number one on the Heredity document?
No, I didn't understand it at all.
I wondered if you guys were gonna maybe just do a little investigation online and figure that one out, but I didn't think so. I'd rather you not. I'd rather me explain it. That's a booger there. It's related to another one in there, and it's like an easy thing to know and just move on. Like I'm going to teach you guys a trick. It's not like a little trick. I'm just going to teach you kind of a rule, and then whenever you see it, you just go, "Oh, I know what this is," and then write it and score a mark, and then get out of the way. So there's certain times that you're taking these exams that there's some questions you just want to 100% get out of the way. Don't waste your time on them trying to figure them out. Know what the answer is just because you can see what the data is being given, and just get that answer out of the way. So there's others that you're gonna have to think about. This one I just want you to have in your back pocket. So I'll show you a little bit of a trick on that one and a subsequent one where you don't even have to think about it. And it's kind of sad because you're not thinking about it. You're just looking at it and just getting it. It's kind of more of a test taking thing than it is anything. But if you're not going to be a biology major, who cares anyway? I'll still give you the backstory. Can't help it. Is it just you two so far? What in the heck? People need to hurry, man. I hope they think that I'm not going to not be here. This class has got a bad habit of coming into class at 8:30 rather than, being a little early. What's up, Becerra? Hey, Becerra, are you in my classroom?
No, sir.
Oh,
I'm at home.
Huh?
I'm at home.
You're at home. Goodness. It's dangerous for you guys to be at home. Playing on your phone, doing whatever. How have you guys been anyway? How was class yesterday?
Classes
For me,
I mean, I feel like we're ramping up a little bit in some of our classes.
Yes, sir.
It's like we're doing the same thing over and over again, but
yeah, we're going to be ramping up in here. It's going to start getting a little more of a fever pitch. Even though this seems like it's going a little slow today, it really isn't overall. I'm trying to push through because we're going to have to finish 2 units this spring. These are 2 long units. I'm going to break this unit into 2 exams. I think I'm going to give your first heredity, like keep, mitosis, meiosis, heredity, one part of it, and then give you an exam on February 2nd. That's when I tentatively have it planned. And then we'll do the rest of genetics afterwards. Oh, there's Landry. Finally, here they come. Landry, did you, how are you feeling about the election? Yo, Landry, how are you feeling about the election? Oh, well, okay, Landry, what office are you running for? Landry, what office? Okay, secretary. Secretary is a communication office. So you ought to use your mic to say something, yo. Come on. Where is anybody else? Oh, here they come. Okay, so you guys should see what items you're going to be needing today. It's pretty straightforward. You're going to make sure that you have copies of this Punnett Squared, the practice, the review, and then the heredity practice. Have copies of these documents available. It's going to be important that you do have these available so that you can refer to them. Hopefully, you have a printer at home and you guys were able to have already printed these. If not, just being able to refer to them as a soft copy is going to be okay. You need to have notes you've made trying to solve these, because the more that you've thought about them prior to class, the better off you're going to be, and you can kind of see where your thoughts were, and then you can hopefully see where I'm trying to push them to go. And then you're going to need paper and pencil for notes to record this information. We'll stay on this screen until 8:33. 8:33 is usually when I have everybody here. If you're here now, please make sure that you guys are coming. I would like you guys to try to log in maybe 8:27 or try 8:28. That way we can start at 8:30. I don't want to lose even 2 or 3 minutes. If we do, we're just wasting other class members' time. We only have 70 minutes to get this done. While we're waiting to 8:33, does anybody have anything they want to bring up? We got 1 minute till I'll start. Oh my goodness. How does my eye feel? Eye feels really bad. It hurts a lot worse today than it did yesterday. I have to have another surgery tomorrow, so I can't take any pain medication because they're going to put me under. I'm going to go through general anesthesia tomorrow, so that I will literally be put asleep on a respirator or a ventilator while they operate on me. So I cannot take a drug that could impact or have an interaction with the drugs that they'll give me tomorrow. So I can't take any kind of pain medication, but pain is weakness leaving my body. I am going to be fine. I got it. I got this. That's right. That's right, kid. That's right, kid. I got this. Okay, it's 8:33. We're going to move forward. There are a couple of roles that I need from you guys today. This is it. So I'm going to be working, and is my camera on? Let me make sure my camera's on. Yeah, my camera is on. I'm going to be working here on this chart paper. I'm going to be working out these problems for you guys. And as I'm working out these problems, I'm not going to be hearing, or excuse me, I'm not going to be seeing the chat. So there are 3 of you guys here. I got Bidu, Morales, Watson, and Williams. I hope that you guys are all here so far. Let me look. Yeah, I got Watson. I've got George, who I just let in. So there's enough here already. Caleb and Morales, hopefully they'll be here shortly. I don't see them yet. But what I will need you guys to do is if somebody puts something in the chat and I do not hear it, for whatever reason, I say hear it, I don't see it, I need you to use your microphone and come on the mic and say, hey, whatever, this person has a question. So everybody's role is to ask a lot of questions. But for these 4 individuals in this class, I want you guys to pay attention to the questions. So if you ask a question today, do not pose that question to me, pose it to everyone. So these 4 individuals can see it, and then you guys are going to use your mics, come on and ask the question for them. Not everybody has a microphone that works. So I think between the 4 of you guys, we'll be able to capture all the questions. If for some reason one of these 4 doesn't see a chat, so doesn't see a question that somebody raised, one of you other guys bring it up. Tech issues. I'm not sharing my screen. I've got my head up my butt for some reason. That's going to be these 3 individuals that say, hey, Pope, I need you to pull your head directly out of your butt. You can't see with both eyes, let alone one. So please, please bring up anything that I may be missing in terms of me sharing my screen function. And then we're going to move on. I'm going to do several things today. First thing I'm going to do is I'm going to this document, and I'm going to stop sharing this screen. And I'm just going to share mine. So I want to pin my video. And as I understand it, when I pin my video, you guys can see this very clearly. At least I hope so. So I'm going to this document, the Punnett Square Review, and I'm going to work on this. I guess it's number 3. I'm going to go to number 3. Okay, so let's look at number 3. So suppose you were an explorer who found a new species of plant. Some of the plants have red flowers and some have yellow flowers. You decide to cross the red with the yellow, and you find a plant that has all orange flowers. What is the probable explanation and why? So let's look at this and see. Let's look at this and see. So I've got red and red. I'm going to put 2 capital R's for red, and then for yellow, I'm going to have, that's the color, that's the phenotype. This is the phenotype on this side, and that is going to equal 2 capital Y's. Just like our blood typing that we finished before, this is a codominant situation. So what you've got 2 phenotypes, red and yellow, and neither are expressed completely. They're actually going to be expressed together. Both are going to be expressed. So let's combine these real quick. So here's again, this is like sperm, just to make sure that we know what we're talking about. These are like sperm. These are going to be my eggs. They're both, in this case, haploid. These are haploid. These are the diploid P generation. We're separating those alleles. We're combining them. Now I've got my F1s. These are going to be my F1s. And all of my F1s here are going to be orange. So capital Y equals yellow, red is capital R, and Y is going to equal orange. So this is very similar to the blood type. And the blood type, you're going to be expressing both of those phenotypes. So before I move on to the next question, can you please pose some questions? Or if that's super clear, we'll move on to the next one. How's that marker, that clarity?
It's pretty good.
It's what again? Speak up really loud.
I said it's pretty good.
Okay. So what is the only possible genotype? Support your answer with the Punnett square. So we did that. We talked about how this is going to be codominance. Support your answer with the Punnett square. That's there. What is the only possible genotype? So the only possible genotype is going to be which one? This one, or this one? Which one are they referring to? Orange, red, or yellow? Yes, thank you, George. You are correct, sir. That is the phenotype that they're, or genotype that they're referring to. Thank you, sir. All right, I'm going to go to the next one. And the next one is going to be a little different. This is where we're going to be working on a dihybrid. So I'm going to run through a PowerPoint really quick before we do this so that you can see how this works. So I'm going to stop, remove my pen, and we're going to go back to my roles. Again, these are the people that are going to bring up questions that I have. Those are you, and then Tech Issues, Landry, Bennett, Miriam, and Brandy. All right, here we go. Not Bennett, what am I saying? Keaton. All right, here we go. So these are going to be dihybrids. These are going to be a little different. I've got the PowerPoint attached. You're welcome to go and check out this YouTube video. I'm going to let you do that on your own. I'm going to walk you through it and how it works for me, or how it works, and you'll just see my explanation for it. But you can come back and you can watch that video as well. So I want to cross, in this case, we've just been dealing with one trait at a time. Now we're going to be dealing with two traits at a time. So this is going to be a little different. It's going to take a little math. It's not going to be too bad. So the first thing we have to do is just find out the genotypes of the parents. In this example, we're going to cross two, the heterozygous individual and another heterozygous individual. Their genotypes are going to be here. Now, I want to teach you guys a little trick, and we're going to come back to this in a moment. There's some times that whenever they give you the genotype, you automatically know the answer without doing a Punnett square. First time we're going to do the Punnett square, then after that we're not going to do the Punnett square. You're just going to look at the genotypes and you're going to know the answer. Simple. So we're going to cross. Here's Dad. They're both what I would refer to as double heterozygotes. They double het for both alleles or both genes, and it looks like fur color is black and white. Coat texture is going to be rough and smooth. So Dad is going to be black and rough. Mom is going to be black and rough, but they both carry the recessive alleles for smooth, and they both carry the recessive alleles for white. Now, I had somebody ask yesterday, why don't we just say black and then have a W for white? That is going to indicate that there would be a codominant situation. They're going to be codominant. These are codominant alleles. These is not the case. Always make the recessive allele the same as the dominant allele, except it's going to be, it's not going to be capitalized. It's going to be a smaller version of that letter, it's lowercase. Okay. You'll have to use FOIL to set this up, and I'll set up the problem up here, but it's a really easy way to do it. It's just really FOIL. It's using first, outer, inner, last. So first, outside, inner, last. So you're going to have to combine these. Now remember, this is a diploid organism. The parents are diploid, but their gametes are haploid. So we're going to go from 2N to 1N. You have what we call independent assortment. When these alleles are, I can pass down one allele and not pass down, and I'm not going to necessarily like have these linked most of the time. So in this case, for this first letter, whatever P is, I can pass down uppercase G or lowercase g with this one. These two are independently assorted from this this first allele, this uppercase P, whatever this is. So we'll go first. So first allele, these are the first when these are paired. And then I'm gonna go outside, first to the outside. So this first one, sticking with this first one to the outside. So you'll go here to here. And you'll pair those and then you're done with this. Then we'll go to the next one. This is going to be the inside and you do the same thing. You're going to go to this first one and then you're going to go to the last one. It's really the same thing as you did here. It's just here to here, here to here. That's it. Questions about this before? You probably have done this. I do see a chat. So those of you guys that are monitoring the chat, feel free to interrupt me. Oh, never mind. It's an old chat. Feel free to interrupt me at any moment that you want to. So let's combine these. I'm going to combine these. So I'm going to pin my video. I think I'm going to pin myself. Maybe I'm going to pin my video. I'm going to cross these booger bears. So let's see. Pin me. And if I make a mistake, then you guys correct me. If I make a mistake, correct me. In fact, if I make a mistake and you come on the chat and somebody sees it, you get 5 points on the next quiz. So let me figure these out. So I've got, and it was G, right? Yeah, it was G. These are my two parent genotypes, my two parents, and I've got to. And you can see there's 1, 2, 3, 4. 1, 2, 3, 4. 4 times 4 is 16. So I'm going to make a bigger Punnett square than what we've been making. It has 16 squares. And again, if I make a mistake and you catch it, that's worth 5 points on the next quiz. Just say so. All right, now. First, enter. So big B, big G. First, and then we got to go to outer. So I went from here to here, just using FOIL. So I've got those taken care of. Now I'm going to go to my little b, here to here, and then here to here. Actually, I should have, ooh, nope. Did anybody catch that problem? Nope. Ooh. Oh, was it R? Was it R? Was that the genotype instead? If it, okay, well, I'm going to stick with G since I've already got it up here. Sorry about that. I couldn't remember. But I already made a mistake. Y'all didn't catch it in time. Oh, okay, here we go. I went to the last. It really doesn't matter as long as you keep it consistent. But here, and then I'm going to go here. So then I'm going to go. The pattern will be the same. Okay, we got to do it again. Oh, this marker is much better. Okay, making sure I don't make a mistake again. Okay, now I'm gonna just combine the alleles. That's all I'm gonna do. I'm gonna combine these alleles. I'm gonna make these a little darker so you can see them a little better. All right, here we go. So and then B. I combine this one and this one. So there, this individual's heterozygous for here. combining here. It's a little difficult to see this with one eye. Ooh, Caleb's here. Uh-oh. Caleb, you say I made a mistake? 1, 2, 3. What do you did, Turner, did you see I made a mistake, yes or no? There's a couple of chats, man. Okay. It should be. I don't see anything wrong with row 3.
I think it's because people are reading your capital G's as lowercase b's or the other way around.
Are you saying right here on the left? This is a row.
Yeah, and
First
that
column, row 3.
Yes.
First column, row 3. She says he's the first column, row 3. So lowercase b.
I'm saying first column, third row.
Oh, first column, third row. Okay, first
That's the
column,
first
third
row. That's the first row.
Yeah, this is the first row, and then the
Yes.
third column.
No, I'm saying it's the first column and the third row.
Okay, first column, third row, right here.
Yes, there's 3 G's there.
Oh my God. Who caught that first? She did. She's right. Who caught that? Who was the first one? Was that Turner? It's the first one I noticed. Yes. Oh, Turner, you got 5 points. +5, baby. You got it. You're happy about that. You're like, what is that crap? Okay, so pay attention. Thank you for paying attention. So yeah, that's a B. Okay. We're good. Boom, boom. I'm gonna go a little faster. Maybe I'll make a mistake. All right, so now I've got this filled out. Everything to me looks okay. Maybe you guys see it a little differently. So I want to look at my genotypes and my phenotypes, and I want to get them in ratios. So first thing we'll do is our phenotype, and then we're going to do our genotype. So let's look at our phenotypes. What phenotypes do we have? And I think if I go back to and refresh my memory, it was black or white. And then it was rough or smooth, and it was R. That's right, it was R. It was not G. How I had G in my head, I'm not really sure. So rough was capital. That will be G. So this will be G for rough. Little g is going to be smooth, which was stupid. And then I got black. This is black. This is smooth. This is rough. And then this was white. So we need to look and see what we get. So this one is going to be black and rough. So black and rough, 1. So I'm going to put black, rough. So there's one of them. So there's 1. This one's black and rough, 2. Okay, it has both of those alleles. That's 2. 1, 2. That's 3. Black and rough, 4. Done with this column, go to the second column. Black and rough, 5. Black and smooth. Okay, so this is a new one. So black, smooth. That's new. So I'm gonna get that one out of the way, just check it off. Black and rough again, 6. Black and smooth, done with that one. Black, rough, 7. Black, rough, 8. Black, nope, this is gonna be black, white and rough. Hold on a second. This is actually black rough. Shoot, I screwed that up. No one caught that? Nope. This is actually black rough. So that's 2. This one is going to be white and rough. Got it out of the way. White rough again. 2 of those. Come over here, black and rough. That should be the last one. Black smooth. Here's my black smooth. Black smooth. Smooth like a baby's butt. That was it. White rough here, and then white smooth. Hold on a second.
You wrote black smooth twice.
Yeah,
so then
I
that
did.
4th one would be white smooth.
Yeah, the 4th one's white smooth. Yeah, I was about to say that makes sense. Okay, so this 4th one here is white smooth. So this one should have been white. Hold on a second. That's white smooth. We already counted one white smooth. No, we haven't. This other one was here. Okay, you're right. Okay, so here we go. So my ratio for this in phenotypic ratio is 9. There's 9, 3, and 1. Now, I already knew that before I figured this out. There's a way to know this. There's a way to know this. Anytime you get 2 double heads, parents that are double heterozygous, in this case, they are double heterozygous for both traits. Do not waste your time doing a Punnett square. If you do, you are going to lose time. This took us several minutes. It doesn't need to take that time. Double heads are always phenotypic ratios of 9, 3, 3, well, in this case, 9, 3, 3, 1. Always. There's only one exception to this, and I'm going to show you the exception, but this is not an exception. If you see this, you know it, move on with your life immediately. Do not fool around. Just get it down. Is the second one black and rough? Yes, it's black and rough. I'm sorry. Okay, so gonna go back to the PowerPoint really quick and let's look at this. I think this is it. I don't have my class. So we're again doing foil, how we set it up. Using foil. And I just did it for you up there. So these are all the alleles that we'll have to use for the Punnett square. And how you get those. You're going to go first, outer, inner, last. So here's my inner and last. Inner, last. We got it. We're set. So what are the possible gametes for it? So we just looked at it. So here's, we just figured this out. The possible gametes were in my Punnett square. That's what we just did. So here they are, sperm. And again, these are eggs. These are eggs. And we got our fur color. We got our texture. And then you just fill them in. Always work with, if you have a dominant allele, put the dominant allele first. This is screwed up. I don't know. I did not actually make this, and I'm really aggravated that they actually had this screwed up. You would need to change that. I always want to put the dominant allele first. If you put them in here, and you can, again, you can see the ratios. The ratios are pretty straightforward. You just go through and you get that ratio. And once you get that ratio, black rough, so my black rough individuals are color coded, the black smooth coat, again, are color coded, white rough, and then white smooth. And you'll see a ratio there that is going to be 9, and me being colorblind, 1, 2, 3, 4, 5, 6, 7, 8, 9. I can see that 9, and then these are too much for me to see. There's between these 6, 3 are one color, 3 are another color, but then you have 1. Always get you're always going to get a phenotypic ratio of 9, 3, 3, 1. Answer the question and go. So there's going to be times on the IB test that if you, and they're not if, they're going to give you a problem like this. They're going to give you a problem like this. When you see it's double het, you answer 9, 3, 3, 1 and move on. Don't bother making a Punnett square. It's a waste of your time. Just have that memorized that if I get a double het, that I'm going to get this ratio. And when I don't, there's another answer that you're going to get. So we've already done this. The other ones, let's move on and we'll get more practice on the guided practice that I've given you. So you can come back and you can look at this PowerPoint as a reference for you, but I want to go back and show you the practice questions and the solution to the practice questions that I gave you guys before. So I'm going to get rid of this, or is there a question before I move on? Yes, no, maybe so. Okay. Away. All right. I'm going to deal with the blood type question. That's here. Now, we did the blood type question before we left. We saw how those actually work. So it says a man on number 5 is het for type B. So the man is het for type B. So heterozygous for type B. So in this case, he's B and O. Another way to think about this instead is this is an I, and I'll actually use this other black marker. That seemed a little darker last time. So IB and I, lowercase I. So this is really going to be BO. You need to set it up this way. They'll want you to set it up this way if they want a Punnett square. And then the female is AB. So the female, she is going to be also het. She's heterozygous, but she's AB. So we'll work out a Punnett square and we'll see what we get. So the man here, and she's A. Ooh, I almost screwed it up. That's an I. That is going to be A, I, B. So their offspring. Done. Now let's see what the question is asking me. So what is the percentage of the ones that have type A? So type A. So type A. Do any of these have type A blood?
One of them does.
One of them does, this one. Yes, so I've got one here. This is going to be, remember that type A can be one of two. You can be homozygous for type A, or you can be heterozygous for type A. Either one. So there is one. So we've got one, in this case, out of 4. 1/4 of these are type A. That's supposed to be type A. And then I've got type B. And I've got one here. BI, remember type B can be BB, can be het, you can be homo for B, or you can be het for B. So I've got 1 and 1, so that's 2/4. And then I've got type O. Are there any typos? I got A and B, that's AB. Doesn't work. AI, that's A, B, B. Nope. So there's 0 out of 4. So this is 0%. This is 50%. And this would be 25%. That solution is that we've taken care of that. We're done with that one. That one's not too bad. These blood typing ones, the codominant situations are really not that bad at all. I like the last question because the last question forces you to really kind of think about things. Like you're going to have to actually spend some time actually thinking about this. So it says a research lab, and I'm looking at 6 as a pure breeding freckle frogs for a cloning experiment. A lab assistant has a pet frog with freckles, but is uncertain whether it's homozygous or heterozygous because it could be either, right? So the pet frog is crossed with a homozygous recessive smooth frog, and half of the offspring are smooth. Okay, so we got to do this. So we got 2 options. We're going to cross both. So the question is the researcher's frog homo or het? So, and we know that half of them came out as being smooth. The only answer is going to be this one. It has to be this one. And let's work out a Punnett square to see exactly why. Actually, it's not. I don't know why I was just saying that. It's actually, it rules that one out. So they're all going to come out as being freckled. It has to be this one. I don't know why I was thinking when I said that. So these are going to be the ones, half of them are like this, and half of them are going to be freckled. So half smooth, half freckled. Smooth, lowercase f, lowercase f, 50% are going to be freckled frogs. That's going to be, in this case, heterozygous. And then I've got 100% freckled, but they're all heterozygous, but they're all going to be het. So this guy must have his pet frog is going to be this one all the time. And, you know, dog breeders and other people that breed dogs, they use this kind of information all the time. I have fish that I use this information with all the time whenever I do it. All right, let's get, that is the last one. Now I want to get to this particular one here. This problem is going to be a little different. So this is your heredity review. Now, how many phenotypes do you see? How many different phenotypes do you see? Taking a look at the data, how many different phenotypes do you see?
Is there 8 different phenotypes?
Well, there are 8 different phenotypes. You're right. So if we were to put those in categories, though, what categories would you put this? I got purple, red, long, and round. So if we put those in categories at the top of it, we have basically here color and what does that say?
Color and shape.
Color and shape. So is this a dihybrid or a monohybrid?
Dihybrid.
Hybrid.
I'm sorry?
A dihybrid.
It's a dihybrid because we're dealing with two different things. We're dealing with color and we're dealing with shape. That's it. So we know it's a dihybrid. When you look at this scenario here, is this a 9331? Look at the data. Is this a 9331 scenario?
No.
The phenotypic ratio is the ratio a 9331.
No, it isn't.
It is not even close. So let's put this in these numbers here together. So I've got purple long. So purple long. So purple long, I've got 4,800, and I'm going to just approximate 4,800. I've got purple round. And that is, I'm going to round to the nearest 100. So that's going to be about 400. It's 390, but I'm just going to round and just get to the nearest 100. Red long, and this is a shortcut, guys. What I'm showing you now is the exception to the rule you saw before. When you see a problem like this, you're going to make an answer and you're going to move on, and I'll still give you a little more of the backstory. It'll save you a ton of time on the test so that you can worry about something that's actually a little more thoughtful and difficult. Here, they're just looking, do you know this rule or not? That's it. And it's really a knowledge-based question. It's not a thinking question. It's just a knowledge question. Red, round. So let's put these into a bit of a ratio. So I'm going to go to, this is 1,100. So let's keep red together, red long versus red round. So I'm going to take 1,100 divided by 400. So 1,100 divided by 400. What is that number? Question mark. 1,100 divided by 400. And I'll ask one of you guys. So let's go to Becerra. Becerra, you tell me, what's this number? 1,100 divided by 400. Hurry up, Becerra, man. 1,100 divided by 400.
2.75.
2.75. 2.75 to 1. So for every one that is red long, you get 2.75 that is going to be red round. Coolio. Let's go over here to purple long and purple round. So I've got 4,800 divided by 400. What is this number? 4,800 divided by 400. Let's go to the most wonderful child in the room. I haven't heard from David Daniel in a while. David Daniel, what is 4,800 divided by 400?
$12.
12 approximately to 1. Now, this is, both of these are kind of an issue, but this one especially tells me something's going on. And it's really what it's called is gene linkage. So this is, I'm going to put this here, gene linkage. Gene linkage. I'm going to get some cards really quick. And I'm going to do a little demo. First thing I need to do is draw this for you. And then after I've drawn it, all right. I'll use a little demonstration to show you why it is the way it is. I'm about to pull my nail out of it. That will be a problem. Okay. So I've got 2 chromosomes. And remember, there's locations for genes. There's locations for genes. And I'm just going to put something hypothetical here. And what do we call the locations for genes? What do we call those? Where we find genes on the chromosome. Thank you, Santos. Loci. Now, if I have another gene right next to it, so let's say there's that G again. Let's use that G. They're next door neighbors. And this is the loci for shape. Let's say this is shape and this is color. And they're right next door neighbors. These genes are probably going to be linked. So as a demonstration, that might help. I've got cards here. Right, so maybe this will help to show you here. So I've got all my face cards. These are all face cards. This is not a magic trick. It's just kind of a demonstration to kind of get the point. And then I've got all numbered cards. I included aces because there's no picture on. Now, if this is a chromosome, and this is a chromosome, and again, remember, you make a chiasma, you go through crossing over, you shuffle your genes. So we'll shuffle these cards. Cards that are really close to one another should actually travel back, because when you're shuffling, you're not shuffling every little single card separately. So if I shuffle these, it's not like every card is going to separate. So let me show you. I'm not tricking you. I've got these two cards. Shuffle them, and then I'm going to pull them apart. There are 2 stacks of 12 because there's only. Now, when I go through these, there's going to be sometimes that the face cards separate and sometimes that they don't. And sometimes these number cards. So I'm going to bet, hopefully, there's a pretty good chance the next card is probably going to be a number card. But let's see how long number cards are next to number cards and how many times face cards are next to face cards. So there we go. Okay, that didn't work that time. It's not going to always be the case. Oh, look, 2 face cards together. Number card, face card, number card, face card, face card, number card, number card, face card, face card. Most of the time, when we just did this, even though we shuffled them, genes that are next door to one another are not going to be separated. These genes that are right next door neighbors will not separate. So if you ever get a ratio and you see, in this case, where we had these genes being so close, the purple and the, I guess, what is that? Purple long, they stuck together almost all the time. So what I'm thinking is these 2 genes are 2 things. They're going to be on the same chromosome, and then B. What's the other thing that you could write for that question? Not only are they on the same chromosome, but they're also what? Because these are chromosomes, but what's the other thing you see here? But they're also, what else should you be able to write for B? Somebody come on the mic, man. No. What goes in that blank? The blank are together on the chromosome. So there's 2 ways that you could get points on this one. And it says on this particular problem, 1 mark. So you might actually have to say both. Thank you, Kimberly Turner. The loci are together on the chromosome. They're very close. So if you were to drop this, we're not hoping for this, but let's say that there is there's a gas leak at my next door neighbor's house, and his house explodes. Is my house likely to be damaged in that situation? Well, of course, his house exploded. Probably going to blow up my house as well, or at least damage it. Do you think it's going to damage the house that's, say, 10 doors down? Probably not. My house is linked to his. And I'll put the rubric for this so you can see all the answers for this particular document, and I'll put it in there. Not yet. I'm going to wait till after 3rd period, but I'll do it today after 3rd period. I'll put the rubric, and you can see the answers to all these problems. All right, I'm going to go to the next problem. And it's a pretty easy one. It says, and they give you a grid, and it's an ABO blood type. It says, using a Punnett square, explain how two parents can have children with different blood groups. Well, is it possible that two parents could have children with all the different blood groups? Now remember, there's different blood groups, right? So we've got, and we're gonna move away. I'll just work this one out over here. So all my blood groups are gonna be A, B, and O. They're gonna be these different blood groups. Is it possible to get those, all four of them? Well, let's see. It is, or not all four, all three of them. It is possible. So let's figure this out. Okay, so I've got yeah. Okay, so I've got here, I've got AB, so this one is going to be AB type. So yes, I've got an AB. Here I've got a B type. That's a B, so this is AB. This one is B. That's type A. And here's an O. So is it possible? Yes, it is possible. It is complete. And not only is it, I mean, it's, if they have sex and have 10 kids, surely over time, because remember, this is just probabilities. We don't know that's going to always work out that way. It's just probability. You have 10 kids, you'll get all of them eventually. I got a question. So what will, yes, this will always be the, if they want you to, and on this particular problem, you can see, I did it this morning when I was drinking coffee. You can see they give you an area to set it up. Now, if they don't give you an area to set it up, personally, I think I would just do it this way. I honestly would just do it this way because it's easier to see for me. I would just say AO, BO. It's just easy to see. That's how I would do it. But if you're going to get points on the IB test, you have to have it this way. They're sticklers about this, and what can you do? They're British. I don't know, the Brits. What are you going to do? Okay, another question. I'm going to skip number 3A and 3B because I'll publish that and they're going to be easy to do. If you look at 3C, 3C is another dihybrid. So let's work out one more dihybrid and then I'm going to do number 4 and then we're going to be done. So let's work out another dihybrid. And I have, and can one of you guys actually read me this question? It's on the heredity. It's 3C on the heredity practice. 3C on heredity practice. Let's go to Rosenbaum. Rosenbaum, can you read 3C on heredity practice to us?
The genes for cystic fibrosis and blood group are not linked.
Ooh, please speak up because I need to hear this.
The genes for cystic fibrosis and blood group are not linked. The parents are heterozygous for cystic fibrosis. One parent has blood group O and the other has blood group AB. Using a Punnett square, determine the probability that their child will have both cystic fibrosis and blood group A.
And it said that they were both heterozygous for cystic fibrosis?
Yes.
Okay, so I'm going to do it shortcut. Is there a place for a Punnett square or is it open on that problem?
It's open.
It's open? Okay, cool. So I'm gonna write it this way just because it's gonna be easier to work out. So we're crossing, there are heterozygous for cystic fibrosis. One's type O, one type AB. So let's set up a Punnett square and let's work it out. Thank you, Rosenbaum. That made my life easier. Foil, CA, CD. Okay, and then that's pretty easy there. CO, CO again, CO. And the only two options I have are little c, the allele for cystic fibrosis, and then we just got to work it out. So we're going to C, A, O. C, capital C, A, O. Capital C, lowercase c, A, O. Capital C, lowercase c, A, O. Capital C, B, O. Capital C, B, O. Capital C, lowercase c, B, O. Capital C, lowercase c, B, O. Capital C, lowercase c, A, O. Capital C, lowercase c, I almost wrote it. Capital C again, A, O. Lowercase c, A, O. Lowercase c, A, O. Again, if I make a mistake, you catch it, you get 5 points. All right, now let's see. What was the question they wanted us to solve, Rosenbaum? They want me to do what?
Determine the probability that their child will have both cystic fibrosis and blood group A.
Okay, so probability the kid is going to be C, C, and A, O. That's it. I don't have any, that's the only possibility. So I'm looking for this. I see it here. I see it here. It's nowhere else. So that's 2 divided by 12, or excuse me, 2 divided by 16. I did the percentage in my head. 2 divided by 16. What is that number? Let's go to Brianna Watson. 2 divided by 16. What is that number? I could reduce it to 1/8. It might help. I just want to know what is 1 divided by 8. 2 divided by 16 and 1 divided by 8. I need the probability. The question asks us about the probability of having cystic fibrosis and type A blood. So what is 2 divided by 16 or 1 divided by 8? I just reduced it to make it easy. What's that probability? What's that percentage? You're correct, ma'am. 12.5%. That's where I was getting that 12. I was just thinking about it. It's going to be 12%. You know, it's half of 1/4, right? So 1/4 is 25, and half of 25 is 12.5%. That's the answer to that one. The last one is a little different. We're going to take a really quick test and let's see what you guys, how you score on this. So let's see. All right, so I'm going to give you a color blindness test. So the last thing you really need to know is sex linkage. All right, I'm gonna take the test. You guys, if you see something, oh, time limit is reached. What? No. Holy crap. Okay, let's do it again. So I see a 2. I see a 6. Oh my God, I see nothing. Sorry. Do you guys see what it is? What is it? Hurry. There's a time limit. Oh my God.
It's 8.
Okay, I see a 3 here. I see nothing. What is it? Hurry. There's
It's
a
a
time limit.
2.
Oh, I don't see it. I see something, but I don't know. What is it?
It's a
What?
4.
4.
I think I said 3.
Yes.
I have nothing. What is it?
That's a 4.
4.
Don't see it. That's a 4, right?
Yes.
Yeah.
Haha. 7. I don't have. I see something, but I'm not sure.
7.
I don't see it. I'm unsure. That's a 5. I see that. I don't know. What is it?
It's a
1.
1.
2. 9.
Yes.
1. That's easy. I have no idea.
That's a good.
4. I don't know. What was
6.
that? 1. Unsure. What was it? Is
9.
it 10? Okay, so 9. That's 2. 4. We got 10 minutes left. I think that's a 5. That's hard to know.
Yeah.
I think that's a 7.
Yes.
9. That's easy. Don't know. 2. 1. I don't see anything at all. What is that?
That was a 5.
I'm unsure. I don't know. 2. 2. 5. Ooh. Unsure. 6. 8.
Yeah.
Unsure. 4. Easy. 1. I don't see anything. Maybe there's something. I don't know.
There was a 5 there.
Okay, so it's view my results. Oh, I have to give my email address. Point is, I have color blindness. Obviously, you could tell. It was pretty tough for me to take that test. So there's something about there's a reason that males have this, or were there any other students that could not see these items either? Because males get this way more than females. Was there anybody else? Or do I stand alone here? Really? Usually there's somebody. Wow. Okay. All right. So it's just me. All right. So I'll show you how this is the line thing I'll do. And this is called sex linkage. So sex linked traits. So we have some ridiculous little chromosomes, and I'm going to share an image with you. Let's see, male karyotype. Let's see, male karyotype. Here we go. Images. I'll go to Google Images is our friend. So let's look. This is a dude. This is going to be a dude. So you can see guys have this, we have 23 pairs of chromosomes. Remember, we have 46 chromosomes total. They're in pairs of 23. The sex chromosomes are our last chromosome, our last pairs of chromosomes. Males are XY, females are XX. What do you notice about X and Y? How are they different between X and Y? What do you notice about them? How are they different from each other? Just visually.
Different from size.
Yeah, the size, that's the big deal. So that is the big deal. I mean, that's the thing that you can tell that's going to be really different about them. So let's go to, I'm going to go to female. I'll put female on here. Female, and this is female human, obviously. It's going to be different for other species. Female karyotype. And let's look at female. Let's see what we get here. So if I look at a female, well, theirs is going to be different, right? So if I look at them, they have 2 X's. Vision, and not just vision, but there's other genetic, there's other traits that we have that fall on the X chromosome. So let's look at what that kind of is like here. So I'm going to go back to me, stop sharing. Females have more DNA than males. So an X chromosome, let's put that as an X, is going to be compared to my Y. So vision, color vision, is found on the X chromosome, and it's found up here at this loci. What do I have to compare that to? So remember, we usually have 2 alleles. How many alleles do I have for vision? So this could be little c or big C. Color vision being the big C. What do I have to compare that to over here? Me personally as a dude, and you guys as a dude?
Nothing.
Nothing, exactly. So I only have 1 gene, 1 allele for color vision. That's it. All you females have 2. You got 2 chances to get it right. I only have 1 chance to get it right. That's it. And that's going to be true for all sex-linked traits. So I want to work out me. What am I? What is Mr. Pope? Okay, so my mom has normal color vision. And how you write this for females genotypes are always going to be X, double X. This is going to be a female. Males are going to be X, Y. Now my mom's, her genotype, I know her genotype because I get an X from my mother. My mother has nothing else to pass down to me but an X chromosome, nothing. She doesn't have a Y. I got my Y from my dad. So my mother has to be a carrier for this. She has normal color vision. She has a normal allele, the capital C. So capital C is going to be color. And lowercase c is going to be color blindness. She's colorblind. I mean, she's not colorblind, but she's a carrier for it. This is, my dad had normal color vision, so I know my father had a capital C, but there's nothing here. Well, let's combine it. And when you combine those, my mom is here. My dad is here. So obviously this isn't me. Let's make that big C. Because I know this isn't me because this is this a male or female? This F1.
Female.
It's a female, right? Absolutely. And there's a little, so this is a probability. Another, my mom and dad could have had a carrier female, and I don't know what my sister is. We'll never know because she's sterile. She can't have children. And okay, so this is one possible combination. Normal color vision. And here. So this is me. When my parents had sex to produce children, there was a 1 in 4 chance that one of them was going to be colorblind. What was the chance of having a colorblind female? Let me ask somebody in particular. Let's go to Landry. Landry, what was the chance of having a colorblind female? Could my parents have a colorblind female? Well, there's a 25% chance they had a female carrier. There's a 25% they had a female carrier. Right here, 25%. This one right here. But she has a dominant allele. So actually, no, they couldn't have a colorblind female. Now, if my dad had been colorblind, sure, but my sister has normal color vision. They both have normal vision, but this one is a carrier for this disorder, if you want to call it a disorder. I don't think have a disorder. I think it's fine. I don't worry about it. At this point, I'm just glad to see. Yeah, but there was a 25% chance that they were going to have a boy with, color blindness. 1 out of 2 of their male children, if I'd had a brother, should have been colorblind. This wasn't me. So I don't know. Let's look and see what my kids are. I think this is cool. So this is me, my doo-doo color blindness. This is Patty. I know nobody else. I'm gonna make this an X. I know nobody else in her family has color vision. So I'm assuming that Patty has both alleles for normal color. So my daughter, capital C, our daughter is a carrier for it. 100% chance that my daughter carries the allele for color blindness. 100%. Blake, 0% chance. His X chromosome came from his mom. All you dudes got your X chromosome from your mom, and you got the Y chromosome for your dad. My daughter, I know, has the gene for color blindness, but she doesn't express it because she's got the normal gene. So I want her to marry a dude that has color blindness so we can get that trait back in the family. I want a little grandson. In this case, it would probably have to be a grandson because we carry them much more often. For a female to have it, I would have to have a colorblind female and a colorblind male reproduce to get a colorblind, or at least a carrier to be able to get a chance at it, to even have a chance. All right, the rest of these problems are on your own. When we come back, I'm going to publish a couple other sets of practice problems for you guys. And then when we come back, I'll answer those questions and we're going to have a quiz. That's it. If you want to come to office hours today, I'll have office hours, and in the office hours, I'll review the very last quiz we had, and then I'll have another quiz, the retake for it. Okay, there's a question. Is it possible for 2 colorblind parents to have a child that is not colorblind? It is not possible. If both parents are colorblind, that means the mother would, in this case, the mother would have to be homozygous recessive for colorblindness, and I'm not heterozygous for anything. I only have 1 allele. So if the only allele that exists, only X chromosome that exists is colorblindness, yeah, all the kids are going to be colorblind. I think it's an advantage. I may not see color the way you do, but camouflage does not work on me. So when I get off,
class is over.
Yeah, I know, class is over. I'm going to put the rubric for these problems on so you can see it, and I'm going to publish another set of practice problems. You guys practice it over the weekend, and then we'll have a quiz. I'll review any questions you have, and then we'll have a quiz. If you want to take the retake from the last quiz, you need to come to office hours today. I'll put a retake up that you'll be able to see then. See you guys at office hours if you're taking the retake. You guys be good.